Hello, this is lecture number 13 in our behavioral neuroscience class, and we're going to be talking about the cells of the nervous system and the great diversity of those cells and the different functions that they have. This is obviously an important part of the field of behavioral neuroscience, is learning a little bit about those cells. And we'll begin that exploration by taking a look at the differences that you see in different cells of our body. These are stem cells that are in the brain, bone cells, blood cells, muscle cells. Take a look at this, fat cells. Take a look at how diverse they are, how different they are, pancreatic cells, and then take a look at nerve cells. And that's what we're primarily going to be talking about. We're going to be talking about both the structure and the function of those nerve cells. So when we begin to explore this, the cells of the nervous system, one of the things that I think is interesting to learn about is just in terms of their numbers. If you take a look at the upper right-hand portion of this slide, you know, this shows our brain and our spinal cord. And the spinal cord consists of about 1 billion of these neurons, cerebellum about 70 billion neurons, and the cortex, the cerebral cortex, about 12 to 15 billion neurons. So there's a lot of them, and they have very specialized functions, as we will learn as we go along. But basically, we divide the cells of the nervous system into two basic types, into what we call neurons, which generally communicate information over pathways over much longer distances, and glia are shorter, and they generally serve a support function. And we'll get into that a little bit more as we go along. But obviously our behavior, whether we're talking about simple reflexive behavior or we're talking about complex behavioral functions, these cells are crucial. The communication that takes place in the central nervous system by way of these cells is crucial for behavioral responses. So a little bit of history. The discovery of cell dynamics was by the Spanish scientist by the name of Cajal, and he really was the first one to begin to describe these individual cells in the nervous system and how they remain separate from one another. He showed that the cells were separate, that they don't really grow into each other. That's how scientists initially thought. Their thinking about the nervous system was very primitive, but it was not until Cajal comes along and he's able, through the staining techniques that he used, in particular, he developed this Golgi stain that you see right here, which really helped us to understand that these cells are really separate from one another, that they don't grow together. So this was a major advance at this time. And again, Cajal was able to do this by way of his examination of cells by way of a simple light microscope. And again, this was a major advance in terms of our understanding of the dynamics of cells. The important components of the cell, again, probably some of this is by way of review from other classes that you may have had, but I think it's important to have these basics so that we have, so that we're talking with the same language. This is an actual electron micrograph of a neuron from the cerebellum of a mouse. And these are artist renditions that you see here of different parts of the cell. And if you take a look, this is again a electron micrograph that you see here. Again, artist renditions that you see in the corners, but here's the nucleus of the cell that you see right here. Here's the artist rendition of what it looks like. You know, this is the outer layer that you see here. This is the nucleolus of the cell. So this nucleus is very important in terms of that genetic material, DNA. It's involved in the hereditary control of the cell. What you see right here are called ribosomes. Here's an artist rendition of what a ribosome looks like. And ribosomes are really important for transporting of proteins and other substances in and out of the cell. What you see right here is the plasma membrane, and that is controlling the exchange of information inside and outside of the cell, maintaining the integrity of the cell environment. We'll talk a little bit about this in terms of the blood-brain barrier a little bit later on. Here's the mitochondrion that you see right here, and this is involved in energy metabolism. So again, these are some of the basic features, the basic components of the cell that you should be familiar with. This is an artist rendition of what these membranes look like, what the cell membrane looks like. And these little green structures that you see here are referred to as protein channels. You know, these channels are going to selectively open up and close and allow charged ions like sodium and potassium and calcium and chloride to pass through them. So again, these are called protein channels, and you see these all along the cell membrane. And when we get into the dynamics of the firing of the neuron and how neurons communicate with one another, these protein channels become very important. So again, here's a protein channel that is open, and again, that opening is going to allow for the diffusion that takes place of these charged ions. Now we have different types of neurons, a few basic types that I want to talk about. Again, probably some of this is by way of review, but I think it's important to have some of these basics down. This is a motor neuron that you see here. The soma cell body that you see right here, that typically is in the spinal cord, and it receives excitation from other neurons and conducts impulses along this axon that you see here. So we're having information then that is being generated in the cell body, the soma. It's going to be initiated in this area that you see right here, which is called the axon hillock. And that information, that electrical and chemical information, is going to be transferred down the length of this axon, in this case to a muscle fiber. And here's the presynaptic terminals that you see right here. This is a covering along the axon. It's called the myelin sheath. These are called nodes of Ranvier. We'll be talking about those when we get to neural transmission. These are the dendritic spines that are going to be receiving information from adjacent cells. So again, these are basic features, anatomical features of the neurons, in this case of a motor neuron. So again, the soma that you see here, this is where this electrochemical impulse is going to be generated, typically in this area right here, which is called the axon hillock. That's going to occur as a consequence of information that is received from the dendrites. Again, we'll be talking about this in more detail as we go along. And that information, electrochemical information, is being transferred down the length of the axon to the presynaptic area that you see right here. Another type of neuron, this is a sensory neuron that you see here. And you can see these sensory endings right here. This is in the skin. Sensory neurons are very sensitive to stimulation coming from the senses, whether it's touch, whether it's temperature, or whether it's odor. And it transfers information, that you see here, to the nucleus of the cell, and then down this axon that you see here to the central nervous system. And again, here's the soma of the cell. So the structure of the sensory neuron that you see here is a little bit different from what you see in the case of a motor neuron. We can generally classify axons in two different ways. We can classify them as being either afferent or efferent. And what you see is the artist's rendition right here of both efferent and an afferent axon. So when we take a look at afferent, and here's an afferent axon that you see right here, that afferent axon is bringing information into the soma of the cell that you see right here. Okay, so the information is going, again, from the periphery down the length of this dendrite that you, down the length of this axon that you see here to the cell body. So that's an afferent. Efferent axons carry information away from a structure. So here's an efferent axon that you see here, which is, again, transmitting information from the soma of the cell down the axon, and again, away from the soma of the cell. So think of afferents as bringing information into a structure, efferents as carrying information away from a structure. So tremendous diversity that occurs in terms of cells in the nervous system, and really neurons vary greatly in terms of three basic qualities: the size, the shape, and the function. So when you take a look at the shape of a neuron, that's really going to determine a great deal in terms of the connections that it makes with other neurons. And the function is really related also to the shape of a neuron. A good example of this is what we call Purkinje cells. We see them in the cerebellum, and they have very, very wide branches. Again, in a single plane, you see these branches going very, very far away from the soma of the cell. So again, a little bit more about this diversity. Again, here's just some examples of this. Here's a Purkinje cell that you see right here. Look at how widely those dendrites, okay, are projecting. Okay, this is a Purkinje cell, and this is in the cerebellum. What you see here are sensory neurons. This is, again, sensory neurons from the skin to the spinal cord. This is a pyramidal cell that you see here, which are generally in the cerebral cortex in the sensory motor area. This is called a bipolar cell that you see right here. We see a lot of these in the retina of the eye, and they serve a very important function there. And this is what's called a Kenyan cell. These are central nervous system cells that you see in the honeybee, and take a look at how much branching that you see here in these cells. So again, enormous diversity in terms of the shape of neurons. Glial cells, I noted to you earlier, they serve a support function. And there are many different types of glial cells that, again, have a great diversity of actions. And let's take a look at a few of them. One type of cell that you see here, glial cell, is called an astrocyte. And this is an actual photomicrograph of what an astrocyte looks like. This is an artist's rendition. And you can see here that these astrocytes are attached to small blood vessels. We call them capillary vessels. And what they do, again, they wrap around the presynaptic terminal, and they take up chemicals that are released by the axon. So again, here's an astrocyte that you see right here. This is a photomicrograph of one. Microglia, on the other hand, here's another photomicrograph of a microglia. And here's an artist rendition of what microglia look like. They're involved in removing waste materials, and these waste materials could be harmful to the neuron. So they serve that very important function of removing waste microorganisms that potentially could cause harm to the cell. Take a look at Schwann cells that you see right here. This is a Schwann cell that wraps itself around an axon. It forms what we call the myelin sheath, which is very important for the conduction of the electrical impulse that is going to be going down the axon. These Schwann cells and oliggodendrocytes, here's an oliggodendrocyte that you see right here. Very important for the transmission of neural information. Radioglia cells that you see right here, very important in terms of what we call the migration process. We'll be learning about that developmental process. It's so important during early brain development, embryonic development, and it's really important in terms of guiding how neurons grow and to what parts of the brain, central nervous system, that they will be going. So again, these are just some types of glia, and again, they principally serve a support function in the central nervous system. Now, the blood-brain barrier, I have this little cartoon here. You know, I have this, I guess you'd call a policeman, and he says, he's responding to these question that's coming from these chemicals that you see here, and the chemicals are saying, hey, we want in. And the policeman is saying, I'm sorry, but you're too highly charged, too large. You're not lipid soluble, meaning fat soluble. You can't enter the brain. So that, I think, captures what we mean about the blood-brain barrier. Okay, it surrounds the brain. It's going to block certain chemicals from coming in. It's going to allow others to come in. The immune system plays an important role here in terms of destroying infected cells throughout the body. But the blood-brain barrier is so important because the neurons of the brain have to be protected because generally they don't regenerate. So blocking viruses, blocking bacteria, blocking harmful things from entering the brain is crucially important. So here's our representation of brain tissue that we see here. Here's a capillary blood vessel that you see here. And generally speaking, large molecules can't cross from the blood to the brain. Again, here's a large molecule right here, and that molecule cannot pass through this capillary vessel into the brain. But there are some uncharged molecules like oxygen, for example, and CO2 that are allowed to readily pass. So again, the blood-brain barrier is serving this very crucial function. So a concept that's important to understand here is the concept of what's called active transport. There are proteins that are going to be crucial for bringing these substances from the capillary system to our brain tissue. things like glucose, hormones, amino acids, vitamins, they are brought into the brain by way of this protein-based system. It's called active transport, and that active transport system is crucial for delivering those basic nutrients to the brain. Glucose certainly being probably the most important one. Our brain tissue, central nervous system simply couldn't survive without glucose. This very simple sugar. That's how neurons are fed. They're fed by way of this active transport system that delivers this very simple sugar to the brain, to brain tissue. So again, this is just some of the basics in terms of the cell. We're going to be moving on in our next lecture to explore a little bit more about the nervous system and how the nervous system communicates in terms of how the cells communicate with one another.